Thank you so much for the invitation to come and share information and my thoughts about salamanders and streams. And I only have like 25 to 30 minutes, and I could geek out about salamanders for a fortnight, which you could probably tell by my use of the term fortnight. So I just love.
I can actually give you up to 40 minutes. So if you go wrong, just keep.
Okay, thank you.
Salamanders.
Yeah, I'm pretty sure I will. So I was given the task of talking about salamanders and streams, and I'm going to focus in a little bit on some specific species that we find here in Oregon, the Torrent salamanders, in a little bit, largely because they're the ones that are going to be largely impacted by some of the new mandates in the private forest accord. So I want to just start out generally, though, just talking about salamanders because and frogs, because amphibians encompass several different groups. So they've got frogs, toads, salamanders, and caecilians. And these are all animals that are very much tied to water. They're most of them are biphasic. So they have multiple life stages and complex life histories that make them really susceptible to environmental change. I started studying amphibians and conducting research on amphibians about 20 years ago. And in that time, I've just become more and more enamored with this taxonomic group, in part because they are adorable. And the evidence is this cute little toad here. And they are also of conservation concern. So we care about amphibians for a lot of different reasons, but part of it is because they are species that are under threat. But before we get to that, I think that we should give a little bit more props to just how cute this species group is. These are toads. This is a Western toad, and right now, and this is an adult, and when they're juveniles, I don't know if many of you have caught juvenile toad emergence up in the Cascades, but they've got little yellow dots on their toes, and when you see that, you can't help but melt, and they are just the best. But there's more than just toads that kind of make all of this worth it. There's also, so there's other animals that live in the Cascades, and I think when I talk about amphibians in Oregon, we have such diverse habitat types and ecosystem types that it would be really easy to just focus on one. And I live in the Willamette Valley, I live in Corvallis, and there's some species that are just really tied to these kind of low valley habitats. There's also a different species group up in the coastal mountains versus the Cascades. So I'm just kind of taking a broad brush right now about what we can find in Oregon. And many of you have seen Pacific giant salamanders. And there's two phases. There's the aquatic and the terrestrial. This is a little terrestrial dude. And I say little with tongue in cheek because they are gigantic and they can scare people. They can actually kind of wig you out because they're so big. And you think, is that a very large fish? No, that's a salamander. This is a picture of the Pacific giant. And this was in, they're all over the Willamette Valley, the coastals, and in the Cascades. But we've got lots of newts too. And the newts are toxic and they're super interesting. A salamander. A newt is a salamander, but not all salamanders are newts. I get that question a lot. We've also got ranids, this group of frogs that we have quite a few of here: Cascades frogs, red-legged frogs, Columbia frogs, Oregon Columbia frogs, and then the bullfrog. That's all the ranids. And they can exist from very high in the Cascades down to the confluence of the Willamette. We've got some. This is the only endemic salamander in Oregon. This is an Oregon slender salamander, and probably several of you are like, why is she showing us so many salamander pictures? But this is a special one. So I just want, this talk is not going to be about the Oregon slender salamander, but I could talk about the Oregon slender salamander. This is the only endemic salamander, meaning that it is the only salamander you can find in Oregon and nowhere else. So it doesn't cross the gorge, and it's just in our Western Cascade mountain range. And therefore, I'm putting it out there right now. I feel like this should be our state animal. Just gonna put that out there. This is an example of watchable wildlife. The Oregon slender. And then we have these really incredible ensitinas and the ubiquitous Pacific chorus frog. We've got red-backed and other plethodons that are terrestrial salamanders. And we've even got this very cool frog called the tailed frog that has little tadpoles with suction cups on their mouths so they can stick to the rocks in these fast flowing streams and not get swept down. They are just evolved to handle our fast moving waters in Oregon mountains. So just a couple of my kind of highlight species of amphibians that we can encounter when we walk around kind of the wetted areas of Oregon. And we're lucky to have as many as we do. The biodiversity of amphibians in Oregon is relatively high. It's not what you would see in the Southeast, down in the Smoky Mountains, but compared to the rest of the lower 48, we're looking pretty good in terms of amphibian species. But overall, amphibians are not doing well. There's lots of reasons why. We've had diseases run through. We've had a ton of habitat loss. We've had climate change, which has desiccated a lot of their wetted area. And in terms of comparing amphibians to other taxonomic groups, amphibians are the worst off. We've compared to mammals and insects and birds. Freshwater fish are pretty bad too. So we almost win the prize for having the most species of conservation concern, but amphibians are hurting. And because of all that land use change and climate change, it hits them in a couple different ways. Amphibians often breathe through their skin. They've got eggs that are permeable. And so if there's a change to the environment, either with toxins or with temperature or with precipitation, they tend to feel it first. So we often use amphibians as kind of an indicator of how the ecosystems are doing. And you'll find different suites of amphibians as you go up these river channels and these streams. And so I'm going to focus mostly on headwater streams in this talk. And that's because the star of this talk, the Torrent salamanders, tend to be found in headwater streams. And it's fun walking up the river continuum. You know, when you get down into the big waters where you've got amphibians that are battling with salamanders and other large fishes, you're going to get very different assemblages compared to when you walk up and you get to these more like different order streams and smaller fish and different species of fish, and then beyond fish. And that's where I like to be. I had this really interesting conversation with somebody in my department, Dr. Brian Sidlauskas, the other day, and he's a fish biologist, very fishy, all about the fins. And we were coming up with this undergrad curricula for people that are interested in freshwater ecology, and everything you put forth had to do with fish. And I was like, dude, there are lots of freshwater ecologists out there who don't care necessarily about fish. And I am one of them. I like to work up in these headwater streams where fish, there's often barriers to movement and fish just can't quite get there. Plus these streams dry up seasonally. So they tend to kind of disappear in the warmer months of the year and then pop back up when that snowpack is there and can replenish the water. And there's some salamanders that have evolved to handle this kind of seasonal shift in water availability, and that includes the torrent salamanders. So I'd like to talk a little bit about torrent salamanders. But first, these headwaters, man, they're so cool because you're walking up and the water's there for a second, and then you keep walking and then it disappears. Then you walk a little further and then it shows back up again. And there's often encroachment from a lot of vegetation in these waterways. The substrate type changes from sandy to rocky to clay. And so it's just highly variable. And these headwater streams are what we're worried about with Oregon, or at least a lot of us are worried about in terms of climate change, because there's some models that have projected that these are the streams that we're going to see less of. So this is a figure that's taken from one of Dee Dee Olson's papers, where she looked at 3 different models that were estimating the amount of streams that were going to dry. So the dry length, the percentage of dry length of these streams in the Cascades. So she was kind of limiting it to Cascade from the headwaters down to the confluence. And a lot of these headwaters are simply going to disappear. And I don't mean seasonally, because that's normal. I mean like forever. So these, even in the, as the snowpack forms, it's not gonna be enough to wet these headwater stream lengths to what evolutionarily we've been used to for the last 12,000 years. So this, you can see that over time, the amount of drying is going to increase regardless of the model that was used. And so this is why we're concerned about torrents and the species that are headwater stream obligates, the ones that are going to have a hard time moving down because of predation by fish or competition with Pacific giants. And the torrents are one of those species groups. And this is a torrent. Isn't it cute? It's just an adorable little animal. And I know I'm anthropomorphizing that, but look at its toes. I mean, honestly, how could you not? And they are, they're biphasic like most amphibians. So they lay their eggs in the water and they hatch out as little baby larvae. They look very similar to this. This is actually a one that's going through metamorphosis. You can see its kind of gills there where its head is meeting its neck. There's a little bit of a barbel there. And that's, but when they're newly hatched, they've got these flaring gills and they're just, it looks like feathers coming off the side of their head, and they are fully aquatic. And then they metamorphose like this little dude, and then they become terrestrial. But even, and so they can breathe on land and they can just get out of the water and be fine, but they're still very much tied to water. In fact, they are the salamander that is considered terrestrial, but still so strongly tied to water that you very rarely find them very far away from a wetted stream length. They used to be just, so they're part of the Rhicotritton genus, and we have several other Rhicotritons in the world, but we've got before there was a taxonomic kind of revolution that happened about 15 years ago, where we just considered all the Rhicotritton in Oregon and Washington to be one species. And then we did some genetics, became a much more viable tool, and we created instead 4 different species of Rhicotritton. So this is these are the Rhicotritton here in Oregon. We have, and I'm just going to use the common names, but on the left we have the Olympic torrent salamander, and they are just found in that Olympic Peninsula. And then right below that species, we have the Columbia torrent salamander, and that's the one. They're very coastal. They're right at kind of the outflow of the Columbia into the Pacific. It ranges across the gorge, so it has populations in Washington and in Oregon, but it's really centered around the Columbia River Gorge. And then we've got the Cascades. So that's the third one from the right, from the left. You can figure that out. So it's the Cascade salamander, and that's the one that's just in the Western Cascades. It does jump the gorge. So we have populations in Washington, but most of the populations of the Cascade torrent are down in the western slopes of the Oregon Cascades. And then we've got kind of the big one, which is the Southern torrent, and that's on the furthest on the right. And this one really is coastal. It's coastal mountains. This is the easiest one to find. I cut my teeth finding Southern torrents just because it's a little bit easier. But probably about 5 or 6 years ago, maybe longer, but this is when I got involved. About 5 or 6 years ago, the U.S. Fish and Wildlife Service said, we're worried about the torrents. They wanted to get a better species assessment for these 4 species, but specifically they were really worried about the Columbia and the Cascades. And that's largely because these are the landscapes that are going to experience the most change in the next 100 years because of changes in precipitation patterns and snow. So the herpetologists in Oregon and Washington, we kind of all got together and we came up with a couple plans on how to better understand the real distribution of these salamanders and what its potential threats are and how to potentially conserve habitat for these 2 species. So this was a project that has yielded some really interesting and important work on and informing the petition to list these two species on the Endangered Species Act as protected. These two species, if you look at them, you're probably like, man, I don't really see a difference. And you just broke up the entire rickettron into 4 species, not sure why, because honestly, even I can't really tell from just morphological characters like what I'm looking at, unless I know where it was found. But it turns out they do have very different genomic structures and habitat requirements. That's what we were finding out. So I'm going to tell you about this species, or this study that we were doing over the last 5 years. The majority of which happened actually in the last 2 years by one of my graduate students, Chris Cousins. So Chris Cousins is a rabid herpetologist. He's very good at finding animals. And so I was like, this sounds like a really good project for you, because torrents are hard to find. Not only do you have to get to the headwaters of the streams in which they live, but you often have to dig down for them, because they can live in the hyporheic zone of these headwaters because they're so seasonal, right? These headwaters dry up and then the water goes down below ground level. These salamanders just travel vertically. So they go up when the water's present and they go down when it's drying, or even when it's not drying. A lot of times you have to dig down to find some of these salamanders. Chris is great at this. So we're like, okay, so we worked with the U.S. Fish and Wildlife Service, we worked with USGS, and we worked with the Forest Service, and we worked with Oregon Department of Forestry. I mean, this was a very big collaborative project that aimed to look at the distribution of these 2 species of salamanders across the historic range, the habitat characteristics that seem to be important. And so we were going to model for that based on where we found them and how many we found at these different sites. And we wanted to test a couple different methodologies on how to look for them. The traditional way of just digging down and walking a lot of wetted stream length and just counting numbers, versus using a genomic approach and taking water samples and looking for a genomic signature in that water that belong to either the Columbia or the Cascade. So I'm going to talk about both of these approaches. The systems that we were looking at. So for the Columbia Torrent, we looked at 30 sites that were distributed across the historic range. And for the Cascades, we again looked at 35 sites. And then it had a structured sampling technique in which we standardized how much time we were out there, standardized how many people were looking, standardized how far we would dig down, just using traditional approaches. And then at 10 sites for the Columbia and 10 sites for the Cascade, a little bit more, we would take water samples. And the water samples were only taken from sites where we knew they were there, where through our traditional like physical searches, we made sure we found them. They were in these stream reaches, and we just wanted to know if they're there, can we detect it with just taking a water sample without looking at all and just looking for their DNA in that water sample. So this was a fun project that involved a lot of modeling. So once we got all of our results and the field crews came in and we knew where they found them and where they didn't, which is really important, plus detailed characteristics of the habitats in which they found them and they didn't, we were able to identify through a regression model that looked at kind of using a Bayesian approach to try and figure out, okay, what habitat characteristics are really important for each one of these species. And so for the Columbia Torrent, so this is the species that's kind of out by it crosses the state lines. You find it in Washington, you find it in Oregon. It's more towards the west part of the Columbia River Gorge. And this is a habitat that's highly modified. There's a lot of urban interface with this habitat. There's a lot of water control There's a lot of potential toxins in these systems. There's a lot of timber harvest. So a lot of these sites were on private land. So we wanted to look at what characteristics were important for finding Columbia torrents. And to be honest, it was pretty intuitive. The habitat characteristics that had the largest relative influence on our models that predicted occupancy and abundance were just basic, water indexes. Was there water there? And how long was the water there? So growing season water vapor pressure type of indices, as well as a topographic wetness index. So the characteristics that really kind of defined the hydrology of the system tended to be the most important. And that's what, as a herpetologist, if you came to me and said, hey, can you predict like what is going to be the important thing for torrent salamanders? I would be like, is there water there? And how long is the water there? And for Columbia torrents, that tended to be the important thing. And here's where it gets interesting, right? Because you've got two species of torrent salamander, and to most people's eyes, they are not different. And yet, when you go over to what is important for Cascade torrents, it turns out to be different. It's not necessarily water so much, it's about temperature. And not just temperature, it's about the range of summer temperatures. So when water's not necessarily there and they're underneath and they're down in that hyporheic zone, the amount of fluctuation of temperature, not just diurnal, but also just over the season, it turned out to be really important for finding them. And so now we've got two species that are of high conservation concern that are responding to two different aspects of climate change. One, water, and the other one, temperature. And they're not the same. So now we've got two different directions in terms of conservation that we can go in terms of like trying to identify refuge habitat for the remaining populations on the landscape. So let's look at the landscape. Let's look at where across the historic range, based on this pretty detailed survey on where we found them, what habitats were important in terms of predicting where we found them, in terms of occupancy and abundance. And if we put that kind of those habitat constraints across the range, what do we see in terms of how suitable is the existing habitat for torrents in these two areas of Oregon and Washington? So this is for the Columbia. And so we've got a habitat suitability model that just is a beautiful color-coded map of where we think these salamanders would likely exist. And so this is the Columbia, or the Cascades. So you can see that the Cascades range is pretty long and thin, and it just kind of covers the western slopes of the Oregon Cascade Mountains. And it pops a little bit over the gorge into the Washington Cascades. And this kind of linear historic habitat range is going to be strongly constricted, and already is, for this particular species of torrent salamander. So the blue watersheds are the ones where we see really good habitat and decent suitability, and where we'd expect these populations of Cascades torrents to do pretty well. But if you notice, there's a lot of red and orange on this map. And this is where habitat suitability goes down, where we're not expecting to find huge occupancy and abundance of torrents, even when we're in headwaters. So these are headwater streams that historically have held these populations, but these are no longer going to be really viable habitats in the long run. And this was a model that had really high confidence based on the field data that we were able to populate into it. Now this is for the Columbia Torrent. So these are the ones on the west side of the Columbia River into the outflow of the Pacific. And again, we see some big shifts in habitat suitability for these populations. And it tends to be the ones on the west side of the Columbia, of the coastal mountain ranges that seem to be most viable. So these are the ones that are going to get more rain. They're where the water is going to stick around a little bit longer. But as you move east, we're going to run into some watersheds that, even though there's headwaters there, those headwaters are no longer going to be viable for populations of this species. And this is based on data where we're looking at right now. This is not necessarily projected into the future. This is like right now, these watersheds are not suitable for torrent. So even though we may have found some in these areas, they either weren't very abundant or they were very sparsely distributed. So when you're working with species of conservation concern, particularly amphibians, handling these animals and really digging down and trying to find them using physical methodologies and traditional survey techniques is problematic. When you've got an animal that you're worried about, you don't want to handle them. You actually you don't want to disturb them. And so we're often trying to identify survey techniques that are non-invasive. Because, you know, when you walk up streams, and I bet a lot of you have actually been on these crews that are looking for some aquatic organism, you know, fish crews or amphibian crews. And what we normally do is we go up and down and we search that habitat. We either use electrofishing, like in this picture, which we do on Rock Creek outside of Philomath, where we just send an electrical current through the water and all the vertebrates kind of like pop up and it just knocks them out, stuns them for a little bit. That's a great way of finding species that you're not necessarily worried about. Or lifting up all the rocks and searching underneath all these rocks and really kind of buffeting the system hard. Or like with torrents, really taking a potato rake and really kind of digging down into these substrates and disturbing the habitat. So one of the things that we did with this study was to try and identify another survey technique that was less invasive, that could utilize our knowledge of genetics as a substitute for actually handling these animals. So we've been using environmental DNA techniques in aquatic systems for 20 years. And the technology has just gotten better and better as our understanding of local genomic libraries have increased, as our ability to work with kind of degraded DNA has increased, and our understanding of just like how we can apply this new technology. So just in sum, this environmental DNA is just DNA that's in the water that animals have shed, right? Because they're shedding skin all the time, or they're shedding urine or feces or blood or gametes. I mean, just think about, you know, when you wake up in the morning, you look at your pillow, all of the skin cells that are on your pillow, we are shedding DNA constantly. And these cells contain lots of different kinds of DNA, nuclear DNA and mitochondrial DNA, and we can capture that. And it's kind of easier right now in the aquatic systems than it is in the terrestrial systems, because everything's kind of contained, you know, so we can just take water samples. So we can go out and we get a water sample and we can filter it through a very small filter that can capture all the DNA that's in that filter. And then we extract that DNA using just basic genomic molecular techniques. And then we run it through a sequencer and we just see what species are present. So we've been using this in Oregon on a lot of projects. I work with the U.S. Fish and Wildlife Service with Brooke Penaluna and Rich Crone, and we've been doing this to index aquatic biodiversity all across the Pacific Northwest. Amphibians are a bit of a pain, I'm not going to lie, because they have huge genomes. 20 times the genome size of a human being. They just are so much DNA. And that can make it a little bit difficult just using a genetic approach, because often you're just like searching through a lot of junk DNA to try and find what you need to identify a species. But I'm kind of getting into the weeds here. I want to kind of broaden back out and just say that environmental DNA offered us a real opportunity to go into these systems, take some water samples and see if it was going to work. Can we identify torrents just by taking some water samples? And can you imagine what a relief that'll be for people trying to, on their land, trying to identify whether torrents are there if these species get listed? So if they're an endangered species, if they're a teeny species, then we're going to have to be really careful with any invasive technique to look for them. And environmental DNA offers a really cost-effective and, more importantly, a fieldwork effective method for seeing if torrents are even in the picture. So, well, before we get to that, so at 10 or 12 sites, actually, I think it was 13 sites for each of these species, when we knew the species was there, we had a protocol for going downstream about 100 meters and taking 12 liters of water. Did we need 12 liters? No, we did not end up needing 12 liters, but, and you can imagine how happy our field crew was with this protocol of going down and into this river system and collecting 12 liters of water in 12 individual liter bottles and hiking that out and all the way to the van and the truck. I mean, that was a very labor intensive type of an ask, but they did it and they did a fabulous job. And Chris Cousins is forever my field hero for pulling all of this off. Planes going by. So got this back into the lab, did the analysis, and I'm going to show you some figures that look like pies. So this is a pie chart. Well, not really a pie chart, but it's a way of showing you the data on how useful eDNA truly was. So this is for the Columbia salamander, right? So the Columbia torrent. And here we have, at each circle is one of our sites. So one of the sites in which was included in our traditional survey, we found torrents there using our physical methods, and we went back and we just took water samples. So we should technically get hits using eDNA at all the sites. So each one of these bullseyes is a site. Each one of the sections, the pie slices, is one of those 12 liter bottles. So each water samples, and there's 12 of them, is indicated by this slice. And each section within that slice, there's 3 sections, indicates a different PCR analysis that we did on each one of those bottles of water. So a very in-depth way of looking to see if eDNA happens. Not only are we seeing if there's a hit at the site, we're seeing if there's a hit at each one of those 12 water bottles, and we're seeing it within each one of those 12 water bottles, like how many times we got a positive PCR extraction of Kesari DNA of the Columbia torrent. So looking at our results, you can see that there's about 4 sites where every single PCR and rep and every single sample of those 12 bottles was a hit. So a ton of DNA was captured using this methodology. And we can say that, yes, with eDNA techniques, we found detection and high levels of detection at a lot of these sites. And some of these sites was a little bit hit or miss. So in the bottom right and then in the middle left, you can see that a lot of those water bottles did not contain DNA. And even if they did contain DNA, not all of our PCR replicates got a hit. But in general, you can say that, man, if there were some salamanders there and we used a pretty in-depth eDNA sampling technique, we found them. So this was a big win. And the Columbia torrent, man, it looks like eDNA is going to work. But I want you to compare that with this. This is Cascade. This is the Rhycotriton Cascades Torrent. And here, keep in mind, every single one of these sites have Cascades Torrents. They were detected, and yet we only were able to detect them in 10 out of the 12 sites. And even in all the samples, only about 50% of those samples was a positive hit. Even less so, a lot of the PCR replicates did not detect salamander DNA across all three. So this was a really interesting result, and we came to realize like maybe something's going on. So just I'm running out of time, so I'm just going to end it there and let you know in conclusion that these collaborative studies are so much fun to do, and they let you really take an in-depth look at species of concern. And in this case, it's the torrent, because they're so important as indicators of stream health. We're more worried about Cascade Torrent than we are in the Columbia Torrent. There's a lot of reasons for that, but partially it's likely going to be more intense summer temperatures that are going to be affecting the Western Cascades. And that's going to affect all the biota in these streams in the summer seasons. And I just want to real quickly, before we pop out to the rest of the program, acknowledge these amazing people. Chris Cousins is the one on the left, and this is really his baby, and he's done some amazing work. And the rest of these folks were really integral, including those two littles on the bottom left, who are my children, and who had to do way too much field work for their for their liking. So with that, I'm going to stop sharing and head back out to Chandra.
Awesome. Thanks so much. I bet they're going to appreciate that time that you made them spend out there in the stream someday. Someday. All right. Let me share mine. Okay. Well, thank you so much for sharing the story of those salamanders and your really cool research. I learned all sorts of things like the genus Rhycotriton. It's a very cool word, and that amphibians have huge genomes. I had no idea. As a reminder, folks, there are a lot of good questions that are already out there. We'll get to those in a minute, but if you have more, just a reminder, you can put them in the question box and we will get to those. I'm just going to talk for a couple of minutes about some of Oregon Wild's work that ties in with what Tiffany's talking about. So first off, if there's anyone out there who's unfamiliar with Oregon Wild, we are a nonprofit organization founded in 1974 with offices and staff all across the state. And our mission is to protect and restore Oregon's wildlands, wildlife, and waters, which means that we've advocated for protecting wilderness areas, roadless areas, wild and scenic rivers, and our last remaining old growth forests on federal public lands for all of that time. It also means that we have advocated for strong protections for native wildlife species, including those on the endangered species list or proposed for listing, as some of the salamanders were mentioned. From spotted owls to coho salmon to some of these cute critters, that's a snowy plover and a coastal martin. And we've also advocated for policies and to support the return of species like wolves and condors and sea otters, some of our keystone species that have disappeared from the Oregon landscape for a time. And while we can all dream of seeing a wolf or a condor in the wild, it's probably more likely that we will see salamanders while we're out and about. These are some that I've encountered in my time. And we definitely work to protect their habitat too. In particular, there's 18 million acres of national forests and forested Bureau of Land Management lands in Oregon that includes some amazing salamander habitat. And that is because groups like Oregon Wild have worked for decades to protect these last remaining mature and old growth forests on public lands and to ensure strong protections for rivers and streams. Our oldest and unlogged forests are our most important for so many reasons, from keeping water cool, which we know now is important for torrent salamanders, to protecting the forests and structures that surround these headwater streams, to also soaking up and storing carbon from the atmosphere in the forest vegetation, the trunks, and the soil. Unfortunately, and despite our many years of effort, there are still some of these forests that are at risk from logging, despite their importance. And today, as we face a climate crisis and have a desperate need to preserve biodiversity, protecting these forests is more important than ever. So we're part of a coalition working to get President Biden and federal agencies to protect our remaining mature and old growth forests across the nation with a durable national rule. And we've made huge progress just in the last year. There are now open comment periods right now, open until June 20th, where you can weigh in on proposed rules that could protect these forests. And I hope you will do so. You'll probably get alerts from Oregon Wild about this very soon if you haven't seen things already, but here's a quick and easy link that you can go to on our coalition page. We're also championing legislation to protect more rivers and streams and the habitat along them under the Wild and Scenic Rivers Act. Senator Ron Wyden introduced the River Democracy Act in 2021, which would protect thousands of miles of pristine waterways in Oregon. The bill has garnered massive support, and we really hope the senator will reintroduce the bill in this Congress as soon as possible so that we can move forward with this really huge opportunity to protect headwater streams and many special rivers that they flow into. So as Tiffany highlighted, salamanders need, especially torrent salamanders, need headwater streams like the one we see here for the water, the flowing water, the water quality, and for these cold temperatures as well. And unfortunately, half of Oregon's forested landscape doesn't have the protections that many of our national forest lands do. So they have a hard time, salamanders have a hard time living in areas where there's extensive road networks and clear-cut logging have damaged these streams and where they have traditionally not been protected. And I'm talking about our private and state forest lands across the state. So for decades, Oregon's logging laws for these private lands have left streams and the wildlife that depend on them largely unprotected. And you can see some of the steep slopes and drainages where there's almost certainly water running at least part of the year in some of these pictures that simply don't have any sort of protection or buffer. And logging rules have given a small amount of stream protection for streams with salmon and steelhead, but not so much to smaller streams. So over the past few years, Oregon Wild and other groups have worked with the timber industry and state government to develop new rules that would better protect streams in general, including headwater streams. So they're in the past, again, very fish-centric, and specifically salmon and steelhead-centric protections have been in place in some places, or on certain size streams where there are fish present, but not so much those smaller streams and headwater streams. And while negotiating the private forest accords, we brought concern specifically for amphibians to the table so that we could see some benefits for them come out of these rule changes. So starting this summer, when the rules go into place, there will be protections for the very first time on non-fish bearing streams in Oregon, some of these headwater streams that salamanders depend on. You can see in this graphic that to move something out of the way, that the previous stream buffers over here on the left, and the sort of dividing line is the fish bearing and non-fish bearing on the left, or on the top. And compared, that's kind of lining up with the new rules that will be going into place. So again, for the very first time, non-fish bearing streams will have a 75-foot no harvest buffer, and then some additional protections for seeps and springs, the real headwaters of a lot of these systems, and would even restrict use of equipment and some other protections on even the teeniest, tiniest streams and some very steep and landslide prone landscapes. I couldn't fit everything into this one slide. So there's a lot that kind of went into this and a lot of rules that are going to be coming into place. And then there, in addition, there's under these new rules, there will be a habitat conservation plan developed that will address conservation of most of the salamanders of concern that are found in this area. So we're really excited about that and really hope that, and there's adaptive management that will be, and science that will be done throughout this process to make sure that these buffers are actually having a benefit for amphibians as intended. And over time, we hope that it will pay off and that these new rules make a difference, or we'll keep working to make sure that they do in the future. So with that, I will turn it off and look at our questions. So I'm sure some have come in since I was talking, but there's a great list of questions that I thought we would start with. First of all, Paul asked a question about whether riparian buffers, and we can talk about this on either federal lands where there's much more protection in riparian areas, or on private lands where they are increasing but still not huge, whether those riparian buffers are adequate to protect the movements of salamanders when they're on land.
That's a great question. So there's been some pretty good work coming out of D.D. Olson's lab with the Forest Service and up in the Washington Department of Fish and Game, looking at the effects of buffers. And there's been some great partnerships between these groups and private timber companies, varying buffer size and doing some good research on that. But they primarily are focusing on riparian and stream dependent species. So not a lot of work has been done on kind of that transition to terrestrial, and vice versa. My lab and quite a few others have been doing a lot of work on just the terrestrial species, the ones that don't rely on streams. That interface and that movement between streams and terrestrial has been a big data gap in terms of research that has happened, and it's pushing forward. So Oregon State University just got management of the Elliott State Forest, which is down in the coastals, and we're doing an installation that's going to be a study looking at that movement from streams up into the more terrestrial zones and then back, because a lot of these amphibians move up in upland habitat and then come back to breed. So the width of those buffers is going to be really important in determining their success. So there's going to be buffer manipulation, there's going to be density manipulation, and we're going to be able to answer that question. But right now we can't.
That's great to hear that that's one of the research questions that'll be going on down there. Yeah, cool. Okay. Somebody had a question about the eDNA sampling, whether you can distinguish between the different species of Torrent salamanders.
Yes, you can. It is just so amazing. But you know what's cool though, because we develop primers for all 4 species and run them all to make sure that we know what's going on. But what's interesting is that you do not get overlap of those species on the landscape, even though their ranges abut each other. So, you know, the southern or the Columbia goes right up against the Olympic, and then the southern goes right up against the Columbia, and then so it's just amazing where there's a distinct line. And I'm really fascinated about the evolutionary barriers that created that dispersal limitation and that kind of species distinction. And so I'm hoping that Chris will stick around for a postdoc and look more into that, because he's really kind of focused on the Columbia right now, and he's doing a pretty hardcore genomics, landscape genetics, and phylogeography to try and understand the evolutionary relationship between the Cascade Torrents and the other torrent species, but really with a focus on Cascade. So I think it's coming down the pipe. But yes, eDNA, man, we can do anything.
That's,
I mean, I'm really excited about this tech.
That's really cool. Kind of related, someone had a question about movement across the Columbia, because the Cascades is in Washington and in Oregon, but that's a pretty big gap. So how did that happen?
Y'all, oh my goodness, this is hot off the press. So with the Cascades, right? So they cross the gorge, and the gorge is really wide there. The genetics show that there is significant population differentiation between the Washington and the Oregon side. So it's been a while since there's been any kind of migration and movement. But Chris's work is identifying that when that movement happened, it was likely around the time, you know, the bridge of the gods, it crosses the gorge, right? So that bridge is named after and reminiscent of a huge landslide that happened in that region. And the tribes have been talking about this for a really long time, about how there was a time when you could pretty much walk across the gorge, and there was just a huge movement of land. And that's when the salamander populations were connected. And so there was a lot of movement then, but that's been, a long time ago. And we would never have known this if there hadn't been traditional ecological knowledge that was informing what we see in these genomic patterns. And it's just the coolest thing I have heard in a long time. Chris just presented this up at the Society for Northwest Vertebrate Biology, and he's about ready to publish.
Wow, what a great argument for really listening to the people who have been here the longest. That's amazing. Cool. There's a lot of questions about eDNA, so I'm going to get to some of those, but somebody asked a question about whether salamanders make noises of any kind, and if there's recordings of those.
Yeah, you know what? If you get on AmphiWeb, which is a online repository for amphibian genomic and life history information out of Berkeley, it's the Museum of Vertebrate Zoology, they've got sounds for amphibians. And salamanders don't make a lot of sounds, but like the Pacific giant will bark. So they make a barking noise, the terrestrial ones. And I just got a hydrophone, throwing it in things. So I didn't know this, but red-legged frogs make underwater noises. And there's so much noise underwater, it's actually really hard to differentiate because the invertebrates are so loud. But we're going to do this in streams too. Its problem is with the water flow, it's also really loud. But we do know that Pacific giants will bark. Never heard a torrent vocalization, but I think that they all have the capacity to do it. It's just a matter of like when and why and how often.
Very cool. Yeah, I didn't expect that they'd be like frogs, like
right.
Yeah, mates, but that's cool that some of them do make noise. We have a high schooler who is asking what you would recommend a young salamanders lover would study.
Okay, so I think forests, right? I mean, just do as much as you can with water and with forests. So I got involved with, there's a lot of local herp clubs. OSU's got a herp club and we love all members. iNaturalist is also a great repository for like learning what's in your region and how to find it. And then hooking up with other herpers and just going out and learning about the life history of amphibians and reptiles is a blast. And then when you go up into the different levels of higher ed, there's specific classes on just herpetology. And it was like the best class I ever took. And this guy was just like Dr. Brad Schaefer, man. And he was just like, let's go on a field trip. And we just go off to a national park and just catch salamanders all day. It's like the perfect job.
We did have a question about how can someone audit your class because you're so excited and passionate and they want to learn from you. So I want to find one about eDNA. There were a whole bunch of questions. So I guess maybe one that relates to your research is how expensive is doing eDNA analysis, and is it sort of prohibitive, I guess, for a lot of labs to be using this?
There's a lot of service labs where you just send your samples and they do the analysis for you. And depending on how much work you're willing to do in terms of filtering, DNA extraction, that kind of thing, the price goes down. And it depends what you want done, what type of genomic analysis you want, just how many markers, how many species you're looking for, all of that contributes to cost. But in general, I would say that with eDNA compared to traditional surveys, the cost is on the back end because it doesn't take a lot of field costs. And by sample, we're getting it down to like $4 or $5 a sample for the simplest analysis. For traditional techniques, the cost is all on the front end because you got to hire your crews and you got to house them and you got to feed them and you got to get them out there for long periods of time. So you're paying for different things, but in the end, it's getting to be about the same.
Does eDNA determine presence, absence, or does it give you a sense of abundance as well? What
a great question. There are some studies that have shown that yes, the amount of DNA in the system is it's highly correlated with the number of organisms that were also in the system, and that was mostly with fish. I've been doing some work on bullfrogs, and I'm not finding in still water that same correlation. But I think that as the tech gets better, we're going to get, that's an endpoint. That's something that we're all working towards, is trying to use eDNA as a way of getting beyond occupancy and detection and and actually getting at abundance. So we do quantitative approaches where we can get at the abundance of DNA, but we need to have a much stronger idea of how much that DNA abundance correlates with that organismal abundance. So we're not there yet.
Yeah. Okay, and I'm going to pitch you one more, and then we should probably finish up. But someone asked about the role of torrents as predators in their ecosystems.
Yeah, they're like a top predator. They're so important. Salamanders are, there's more biodiversity of salamanders and amphibians in these systems than there are breeding birds, than there are mammals. I mean, there are so many salamanders on the ground. I could go off for days, but they are a huge predator in this system. They're a top predator in the streams themselves, but they're also an important prey item for a lot of avian predators, a lot of snakes, a lot of small mammals. There's lots of things that eat amphibians, and amphibians eat a lot of things. So they are a key component of these food webs and so important. And so if they disappear, then that's a huge loss in these communities.
Kind of like, I don't know, maybe we don't even know, right? But like we talk about sea otters and the role top predator that they play. And these are just on like a different scale and a different habitat.
Yep, it's the exact same thing. And they kind of, what they eat, if they eat a certain type of shredder or a certain type of decomposer or a certain type of invertebrate that has a different interaction with the carbon sources in these types of environments, then I mean, the climate change implications are actually huge for their total overall impact.
Well, we will all stay tuned to learn more from further research. Thank you so much for sharing all of this great information with us tonight. And for those of you, again, who maybe joined late, we will have this recording available tomorrow on the Oregon Wild website. And thank you so much, Tiffany. We really appreciate it. See you all.